This is the second video for this week, and this one is entitled Dose Response. Video 1 this week was about linear regressions. Part 2 is on dose response, which is a very similar idea, but it uses linear regressions to take an authentic look at how you could use a bioassay and regression analysis to look at real-life biological questions, quite often in toxicology or in the field of pharmacology. In dose response, we're asking whether or not we can find a physiological or biological effect over a range of dosage concentrations, maybe for a toxin or some kind of therapeutic drug. And we're looking at a regression analysis of those response values, and they often show what's called an S-shaped or sigmoidal curve, which is somewhat similar to enzyme activity. Let's break down a model of a dose response curve. And I think for the sake of this presentation, we'll just focus on a dose response curve for a toxin, meaning we're looking at some sort of negative response based on increasing concentrations of a chemical. So like most graphs, we have an X and a Y axis. And on the X axis, we are looking at the dosage of a drug that we are testing, increasing from 0 up to some upper limit. And on the Y axis, we are looking for a response as a percentage of a test population. We're looking at dose responses, and in the field of toxicology, we're trying to establish how much of a population is affected by a drug or a compound at a particular concentration. So if we thought about how this experiment might have been carried out, we would have a population of 100 test organisms that were given 0 grams or 0 milligrams of our drug. And at the end of the experiment, we would score every one of those individuals and say how many out of the 100 are showing signs of the response that we are currently looking for. And that response could vary based on our question, but it might be lethality. In toxicology, we're interested at what concentrations become toxic in organisms. We could be looking at carcinogenicity, or the tendency to cause cancer in cells, tendency to cause heritable DNA changes, or mutagenicity. Or if we were looking at therapeutic drugs, we might be looking at reduction of symptoms, right? But as we move up the scale, we're going to give another population of 100 organisms 5 milligrams of the drug, and over a period of time look to see how many of those are showing symptoms or our response. And as we move up the line, we start seeing that more and more individuals from our test population are starting to exhibit symptoms or exhibit the response, to the point here in our example where at 35 milligrams, at the end of our experiment, 100% of the organisms in our population are displaying symptoms or showing the response that we are testing. If we're looking at the toxicity of a substance, we would be looking at the lethality. We would say that 100% of our organisms died at a concentration of 35 milligrams. This brings up another important benchmark for all of our dose response studies, and that's the 50% mark, or the concentration at which 50% of the members of a test population exhibit symptoms. For lethality, we call this the LD50, or the lethal dose 50, in which the concentration of our toxin results in the death of 50% of our test subjects. Now, just because I think that this is pretty neat and interesting, here's a list of some of the LD50s of a number of different toxic substances. And in the experiment that we used to collect this data, we were looking at the lethal dose in mice or rats. So looking down our list, I think there's a couple of these that are probably pretty familiar to you. Sodium cyanide. Working down, we got nerve gas. First interesting one that you might not have seen before is tetrodotoxin, which is pufferfish toxin. Some people like to eat risky sushi. Pufferfish produce this neurotoxin in a number of their organs at very high concentrations, and people will eat sushi made from pufferfish because it's risky and it's a little bit exciting. And if you were to get a little bit of this tetrodotoxin in your food, it will cause your mouth to become kind of tingly, but as it starts working through your system, if you get a lot more of it, it can paralyze your muscles and your diaphragm. And of course, your diaphragm is responsible for helping you breathe. So the more of this toxin you ingest, the more possibility you have of paralyzing your diaphragm, and eventually you could die for that. But we see that the LD50 of tetrodotoxin is fairly low. You only need 0.1 milligram per kilogram to kill 50% of your test population of mice or rats. We can go down to some slightly more toxic substances like Agent Orange. Dioxin is a byproduct of the production of the Agent Orange that was used in Vietnam to defoliate trees. That's toxic at a rate of 0.003 milligrams per kilogram. Even more toxin than that is ricin, the toxin that's extracted from castor beans, and you might have seen this in Breaking Bad. It was one of Walter White's favorite compounds. But this guy down here at the very, very bottom of the list, this is one of my favorites. So let's look at this one in a little bit more detail. Botulinum toxin. Tetanus toxin A or botulinum toxin A. So we'll look at this one in a little bit more detail. Clostridium botulinum is a soil bacterium. You're going to learn all about bacterium in the next 4 weeks of lab, so let's get started right now. Clostridium is a gram-positive bacillus, and it forms resistant spores in the soil. It lives mostly anaerobically in the soil, and it produces multiple toxins, one of which was the one that we had on our list on the slide before. Botulinum toxin is currently believed to be the most toxic compound known to humankind. And animal models suggest that if you were to inhale 0.7 to 0.9 micrograms of aerosolized botulinum toxin, that would be sufficient to kill a person weighing 154 pounds. So we're talking about tiny, tiny quantities of very toxic stuff. So how does it work? Let's look at the mode of action. So what you're looking at right here is more than you need to know for this course, but this is a neuromuscular junction. And your neurons produce vesicles that contain acetylcholine, which is a messenger molecule. And your muscle cells contain acetylcholine receptors along the membrane. And when a neuronal impulse happens, it will release some of this acetylcholine, and it will bind to the acetylcholine receptors, and then your muscle will respond in some way. It will contract. Botulinum toxin blocks this process so that when a neuronal signal happens, acetylcholine isn't released, and because there is no muscle release, that means that there's no subsequent muscle contraction. So the two of these have been uncoupled from one another by botulinum toxin. And I think you can imagine how the lack of muscle contractions could be a negative impact on somebody that's living. Going back to the puffer fish, your diaphragm will stop moving, it'll stop contracting, and you will eventually stop breathing. Is there any possible way that botulinum toxin, the world's most toxic compound, could have any therapeutic benefit, right? It causes muscle paralysis, so it's got to be bad, especially at very, very low concentrations. But it turns out that the dosage is what makes something a poison. Botulinum toxin type A is quite literally the active ingredient in Botox. And we've all heard of Botox before. You've heard about the happy Botox face, and the sad Botox face, and the excited Botox face, furious, confused Botox faces, all of which look identical to one another because Botox is a muscle paralytic, and it paralyzes the muscles underneath the skin in your face when it's injected, which helps reduce those worry lines or the wrinkles across your face. Botox is a perfect example of how the right dose differentiates a poison from a remedy. So again, here we've got our bottle of Botox, and this commercial bottle of Botox contains 100 arbitrary units of our toxin, right, to be used for therapeutic means. If you've got crow's feet around your eyes, some wrinkles, 6 to 20 units of these injected into your skin will help get rid of them. Just like 20 to 30 units will help get rid of the worry lines across your forehead. And unbelievably, if you're too sweaty, an injection of 50 units of Botox underneath each of your underarms is sufficient to stop your perspiration. And out of curiosity, I pulled out and I looked at the instructions for Botox that's provided with each of these boxes, and 1 unit of Botox corresponds to the calculated LD50 in mice. Right? Now that you know what the LD50 is and you know about dose response curves, you can recognize that just 1 single unit of Botox is enough to kill 50 out of a population of 100 test mice in the lab. So that's quite a lot of toxin that's being used to paralyze the muscles underneath your skin to get rid of all of those wrinkles. Botox is not just for the vein. There are other ways that it's used as a therapeutic. I'm not sure this is even still on NPR right now. I should probably check. But one of NPR's radio show hosts was named Diane Rehm, and she has a condition called spasmodic dysphonia, in which her vocal cords will spasm uncontrollably at times, and that affects her voice. And she's received Botox treatments, in which she's had Botox injected directly into her vocal cords to partially paralyze them to reduce that spasmodic dysphonia. And it worked very, very well for her. Let's get prepared, not for this week, but for next week's lab, in which we start microbiology. We're going to be using Bunsen burners, and we're going to have bacteria and chemicals out on the bench. Everybody is going to be required to wear goggles. And if you don't have them, you can't come to lab. So I'm giving you now a week to go out and find some. They can be standard goggles. Any of these shapes would work, including the big wraparound elasticized goggles that make you look like a raccoon after wearing them for 3 hours. Again, we're going to be using them in lab, and if you don't have them, we're going to ask you to turn around. In other semesters, we've had extras, but right now, with health concerns, we won't be sharing goggles. And here are your final announcements for the rest of the week. Quiz number 5 opens up at Monday at 12 a.m. and it closes Tuesday at 11:59 p.m. for a total of 48 hours. A lot of people have been missing these, and they are a big part of your grade, so please remember that you have a quiz. This quiz is going to be 25% relevant to our Daphnia labs. It's going to include some review. Make sure you do have a calculator, and remember how you interpret graphs and analyze regression equations. In terms of assignments, you have a diffusion lab report part 2 that is due in lab this week. Please make sure that you are reading the instructions and you're following the rubric on Canvas to make sure you're answering all of the elements of your lab report that will be graded. Your Daphnia assignment part 1, which are the questions from the in-lab part of the lab, and 5 extension questions are also due this week to your faculty member or ULI. The last experiment that we're going to do in Daphnia, creating our dose-response bioassay, is going to be due the week of November 2nd. There's also a Daphnia bioassay write-up grading sheet that establishes and tells you all about all of the elements that are going to be graded in this upcoming lab report. So make sure you're looking at this and you are clear on what it is that's expected of you for your reports. Have a good week.